Okay, sure. Again, you guys can turn on your video if you wanted to make more interactive, and you can turn on your audio if you want to ask something, say something.
I think once the session starts, we would request the audience to switch off both the audio and video, and they can join at the end. That would be when the session starts. One of the things which you can tell people is that let them mute their video and audio, and then put the questions in the question answers. And then once we start the discussion, I think we would let them in. That would become easier. Yeah.
Okay, we are live now.
Okay.
Yes.
Sure.
Let me turn off my video, so we can go ahead.
You can be.
Okay, all of you, can you see my slide? Can, right?
Yes.
Okay.
Yes.
Good morning, people in India, and good afternoon, Singapore. So thank you so much for joining us today, Sunday forenoon and afternoon. Thank you so much, really. Simple housekeeping. So as Prof just told, you guys can turn off your video and audio for now until we start the panel discussion. But you can very well start asking questions in the chat. Let it be specific to the speakers today. Neha is a biotechnologist who's also a bioinformatist, and Bilge is a biochemist and biotechnologist. So you can start thinking in that line and ask questions related to career or future prospects of biotechnology in the chat session. But if you have questions beyond today's session, you can write to us at avantgardeinitiatives.com. It is so complicated, so please take a picture or screenshot of this page, right? So how it goes today is, I'll just give you a very quick look into Avantgarde Initiatives itself, and I'll pass on the session to Prof, who will introduce the speakers, and the speakers will talk about themselves and their research for 5 minutes each, after which we will have a panel discussion for up to 30 minutes, where Prof Sai Baba, who is our moderator or chairperson today, he is from the National Institute of Advanced Studies. So he's a very, very nice guy. He's an expert in science communication, HR related stuff, and also policy making. So he's also the to-go person if you guys have anything, any doubt or any queries in this field. So after the panel discussion itself, we will open the question and answer session for the audience. It would be really, really great if you can unmute yourself, and also, if possible, the video. You can turn on the video and ask yourself. That would make it more interactive. In case you're shy, then we will take your questions on the chat and I'll ask on your behalf. And I will also introduce a little bit about the mentorship program that Avantgarde Initiatives has started, which I think would be very useful for the audience today. Right, so that is it about today. Thank you again, all of you, for joining. A little bit about Avantgarde Initiatives itself is it is a one-stop platform for life skill-based education matters. So it is for anyone who wants to, who believes in empowerment through education. So when I say anyone, it's anyone. It doesn't matter whether the student is from a premier institute like in Singapore, NUS, or IITs in India, or any arts and science college. We make sure that all of them get the equal and sustainable opportunities. So for example, today in the audience, like you are more than 30 people, but different various other backgrounds. So virtual webinar has made this possible, right? So this is what we do even beyond the virtual workshops. Even in reality, when things get back to normal, this is what Avantgarde Initiative does. We provide career guidance, research mentorship, personality development stuff, and global connections and networking, all for anyone who seeks it, right? And all of us, whether it's me or Heman and Bilge, the speakers, Bilge, Neha, or Prof Sai Baba itself, we all do it on pro bono basis. We are not getting paid ourselves for doing this. So that's one thing that I would like to highlight. So so it's about people who are passionate in empowerment. So as I said, we are global, and we are also there is no gender, race, or socioeconomic barriers. So we do the programs for everyone, and we have different suite of programs. We don't have time to talk about it now. So please go to avantgarde-initiatives.com to have a look at it. You can take a screenshot of this. Avantgarde, I know the name is so confusing. Avantgarde means anything that is out of the box and progressive. So that's why I named it Avantgarde. But just take a screenshot or photograph of this page. And I'll be talking about this program later, but just a quick look into it. We have a new program collaborating with NUS MBA. You know, NUS MBA has one of the highest world rankings. So the students and alumni of NUS MBA are our mentors on board who are willing to give mentorship for mentees, any one of you in the audience today, for free, of course. It's a 3-month program where you can have an one-on-one interaction with the mentor anytime at your convenience, and they are going to help you with career, personality development, and also entrepreneurial skill development, right? So I'll talk more about this program later. And today we have two of the, my very, very closest friends, Bilge and Neha, who are biotechnologists, who are willing to share their insights into the future of biotechnology. Over to Prof now. I think he will take it from here. Yeah, bye.
So thank you, Viveka. So good morning, India, and good afternoon, Singapore. And we are here now on the 4th session. This is an initiative avant-garde webinar and panel discussion on the theme of innovation in the times of crisis. And it was started on 14th, and we are 4th session. It is spearheaded by Viveka, which you could see from her introduction. So I take this opportunity to welcome all of you to this session. And we have Dr. Neha and Dr. Bilge with us who would share their experience. And Viveka and I would be hosting and moderating this session. And I think the theme today is on innovating biotechnology for the future. I think I very briefly, Dr. Neha is a research associate with A-Star at Singapore. She wants to call herself a very passionate researcher, driven to understand different systems using data analysis. And Dr. Bilge is a research fellow at Nanyang Technological University. I think she's an experienced researcher with a demonstrated history of working in the higher education industry. You can see the diversity and the vast experience with young research that we have. So let's start. I think as Viveka told, initially we'll ask the panel members to introduce themselves the way you feel appropriate. And then after the introduction is over, then we can go to each of you to start. And then I have a set of questions to ask you. We will continue the interaction like that. So Neha, so you can start.
Okay, so hi everyone. Thank you for having me here. And like this initiative is very close to my heart, like empowerment through education. So thank you very much, Viveka, for including me on this. I'm a research associate, as Prof said, at GIS, Genome Institute of Singapore, which is a part of A-Star here in Singapore. My work mostly involves currently computational biology. So it's just answering biological questions using computational techniques or mathematical models. So currently I'm working on cancer metabolism. So we have a lot of cancer data, so patient data, sequencing data from patients and how their disease progresses and all that information. And we do analyze that data to get to understand how cancer is different in different people, or how is it similar in different people and how we can target it differently. Apart from that, before that, I did my PhD from Iceland. And there also I was studying metabolism. So I've been working on metabolism for like 8-9 years now. But there I was studying metabolism of cells in culture. So how cells adapt to growing in culture where they have a lot of metabolites. So basically we are all eating, even the cells, all individual cells are just eating and dividing and growing. So I've been studying that. Yeah, basically before that I'm from India. I worked in Jawaharlal Nehru University for some time. And I think that's my introduction. in short.
Yeah. Thank you, Neha. And can we now ask Dr. Bilge to introduce herself?
Yeah, sure. Yeah, my name is Bilge, and as Prof said, now currently I'm a research fellow at Lee Kong Chian School of Medicine, which is under Nanyang Technological University. Now technological universities have School of Medicines also. Yeah. And so I have few slides to share just to summarize my journey and you know, how I came and what's my research is about very briefly. If you, yeah, can you see my slides now?
Yeah, we can.
All right. So, okay, yeah, as I said, I'm a postdoctoral researcher at Lee Kong Chian School of Medicine, and I'm from Turkey. So I'll just briefly talk about my journey, what I did, the education, and then how I ended up being in Singapore. So, so I started my education in Turkey. I got my Bachelor of Science degree in chemistry. And the education we got was actually very broad. So they taught us, I don't know, currently I think people realize that they need to teach specific subjects, but at my time in 2007 in Turkey, actually there was like Department of Chemistry and you are learning every single subset of chemistry, which includes quantum chemistry, physical chemistry, biochemistry, etc. And at the end of my last year, so I asked the question to myself, so which subject interests me most, since we learn all of these stuff. And my heart was always saying that biochemistry is very interesting. It's the, you know, how life uses molecules, how nature uses molecules to create life. And, but if you look at my grades, it was polymer chemistry. So there was this discrepancy between what my heart says and what I can achieve, or at least at that time it looked like that. But the one important lesson that I learned here was mentors are very important in our lives, because the reason maybe I didn't push forward in biochemistry, or I didn't take you know, extra mile to do better in biochemistry was because my teacher was not that inspiring. On the other hand, if you look at polymer chemistry lecturer and the labs that they, you know, designed, the experiments that they incorporated into curriculum was very interesting, very engaging, and you know, like real life examples. So it was, I was like, okay, you know, very interesting, very inspiring. So I will take, you know, extra effort to ace in this subject. And since I graduated from Bachelor of Science, I got my degree and I got scholarship in Master of Science in Material Science and Engineering, like Rebecca's PhD subject. It was about polymer chemistry because of what my grades say that I can, you know, achieve in polymer chemistry. And the project that I work on was an industry collaboration. So we created these anti-icing coatings. So in Turkey, it used to snow a lot. Now, because of global warming, actually, the time that, you know, we get snow is maybe 1 month only, but previously it was quite, you know, harsh winters. So that was my main project, creating these anti-icing coatings using polymers and nanoparticles. But my supervisor also gave me a side project, which was about thermal characterization of polymers that have similar chemical structure to proteins. So protein biomimetic polymers. And while I was reading the, you know, articles about these, of course, I stumbled upon, you know, research articles related to hardcore biochemistry and protein science. So this side project actually revived my, you know, interest in biochemistry. And then at the end of my master's, Master of Science degree, I said, okay, I want to do a PhD and it should be related to, you know, biochemistry. I want to learn the interplay between protein and lipids. At that time, it was already in my mind. And in 2013, I came to Singapore to do my PhD with A-star scholarship. And what my PhD project was about, characterization of protein machinery important for maintaining lipid asymmetry in bacterial membrane. So it's like a very complicated name, but basically, so you may ask, what is lipid asymmetry in bacterial membrane? So if you look, I'm sure you all know about bacteria, and the most famous one is, can you see my cursor? The most famous one is E. coli. So this is artist's illustration of E. coli bacterium. And if you zoom in its membrane is like separating bacteria inside of bacteria, which is cytoplasm, from outside. And if you look under cryo-electron tomography, you can see there are two layers in bacteria. So one that is close to outside. So this is inside bacteria, which is called cytoplasm, and this is outside. And there are two layers. The one that is outside is called outer membrane, and the one that is inside is cytoplasmic membrane or inner membrane. So if you look at outer membrane structure, which was depicted here as a cartoon, it has asymmetric lipid structure, which means the lipids at the outer leaflet of the outer membrane is mostly lipopolysaccharides. And the ones that is in the inner leaflet is phospholipids. So this lipid asymmetry is very crucial for bacterial survival. Because of this lipid asymmetry at the outer membrane, the antibiotics, the small molecules that we try to use against bacteria has difficult time to enter the bacteria. So if you have compromised outer membrane lipid bilayer, your job is easier to kill bacteria. So time to time in bacterial membrane, these lipids in the inner leaflet spontaneously flip back to outer leaflet, which now you have compromised lipid asymmetry, right? So you don't have asymmetry anymore, it's symmetrical. And there is a machinery that senses these aberrantly localized lipids and moving them from the outer membrane. So I characterize this machinery in my PhD in molecular details, how these proteins are interacting with these lipids and doing their jobs. So you may ask, why is it important? But I already gave the answer. So we are having COVID is aside, we now have actually another problem, which is antibiotic resistant bacteria. And we need to have a new antibiotic targets that we can, use new antibiotics. So this system might be proposed as one of the new targets. So that was my PhD. So 2 years ago, I started my postdoctoral research in Lee Kong-chian School of Medicine. I like bacteria, but I was thinking maybe I can also, explore mammalian proteins and lipids, actually lipid interacting proteins in mammalian systems. But the actor that I picked was quite different than the lipids that I studied before. So bacterial lipidome is quite simple, but mammalian, including humans, obviously, is very complex. And cholesterol is very important lipid in our bodies. And bacteria doesn't have the cholesterol. So that was something new for me. And cholesterol trafficking in our cells are very important. If there's any improper cholesterol trafficking happens, it can lead to several pathologies that I indicated here. So to give an example, Niemann-Pick disease type C is one of the disease that caused by aberrantly localized cholesterol in our lysosomes. If you remember from your high school, college subjects, we have different organelles, mitochondrion, lysosome, endoplasmic reticulum in our cells, right? So lysosome is the place that unwanted things being degraded, and also the place where cholesterol arrives from outside. So if you have this disease, you have a non-functional protein, MPC1, that cannot export cholesterol from lysosome to the other parts of the organelle. And that leads to, shortened lifespan, also developmental problems in people. Another disease that caused by improper cholesterol trafficking is atherosclerosis. I'm sure you have heard high cholesterol, right? Everyone has at some point high cholesterol in their body, in their blood, and it causes heart diseases. So why it happens is because we get cholesterol from diet, and you have a receptor in your membrane, plasma membrane in your cells. But if it doesn't function properly, you cannot get cholesterol from outside into inside your cells, and it accumulates in your bloodstream and eventually clogs your arteries and causes heart diseases. And also there's a new research going on that's showing cholesterol trafficking in brain is very important, and in its dysfunction, it may lead to even mental retardation and other diseases. So actually I already explained why is it important, right? Why is it important to study all these basic research and the interplay between lipid and proteins? Because if you don't understand how they do their job, when they stop doing their job, you can't find solution for them. So yeah, that was like, I tried to really, switch everything that I tried to do in the last almost 10 years. So if you have more, questions about my research, you can type my name, Google Scholar or LinkedIn. You can, ask me questions on LinkedIn. And I try to explain. And if you have again questions about if you see similarity between what I did and what you are going to do or what you are doing, and if you want some advice, you can also ask me, send me a message. Thank you.
Yeah, thank you, Dr. Bilge. Actually, you made my job difficult by explaining very well. Some of the questions which I thought I should ask you, you covered it, but I will innovate myself to, yeah, I'll ask you. Yeah, so now let's start. I think now the theme of the, maybe we should request the people to, yeah, so I think, the theme is on innovation in the times of crisis and also in the new norm. I think the first question is to both of you. I think I would like to ask you, we are discussing innovating biotechnology for the future, right? I think this is what. So I would like you to firstly comment on innovation. When will you say it's innovation, and when would you say it's a research and innovation? When is that would happen? And also how it unfolds in the domain of biotechnology or broadly in bioscience research. And we discuss, is it biotechnology, biochemistry, or whatever, bioinformatics. I broadly call them as biosciences, but how it unfolds in this domain. So Neha can take a shot at this question, then followed by Dr. Bilge. Okay.
Okay, so I think like innovation, like its definition is creating a process or a product which adds value. And it's the same for biological sciences, like you say, and it could also be an improvement over like
no, Neha, I have to interrupt you. See,
yeah.
Doing research also is that, no? Any research is
Yeah.
that only.
So research and innovation,
are they same, synonymous, or something more? You just have to tell me, is it same or it is?
I think research is broader, I would say. And so research can be basic research as well, right? So we can study about like cellular systems, but maybe they are not like relevant or applicable right away to add value. But in a broader scale, in a longer term, in maybe like next 10 years, where there's more research and more knowledge is gained about the whole system, that's when innovation comes in. So innovation is maybe, I would say, end product, like, or a later product of research. So research, like basic research accumulates the knowledge, and with that knowledge, people can innovate, is what I would say. And also in biotechnology, if you think like right now, biotechnology or biochemistry or biophysics, like all these fields, There are different subdomains of biological sciences. These are just application of technology, like different technology or different knowledge, like knowledge of chemistry, knowledge of physics, knowledge of computational programs, algorithms, maths, statistics. So all that applied to answer biological questions or solve medical problems or biological problems. So I think innovation in current biological field or biological science, it comes from innovation in technology. So for example, like innovation in sequencing, in DNA sequencing in the past 10 years has helped us a lot in health sciences. So now doctors can sequence individual patients. And I know for example, in cancer, it has helped a lot in the treatment or management of cancer. So what I'm trying to say right now, so it's both. Research is in the background, innovation is in the forefront, for example.
Thank you, Neha. I think what you are trying to convey to us is that you have results, you have research, results are there,
Yes.
and there are needs for the people to solve, and then combining them in a special way. I think combining, ultimately every research is aiming at solving a problem. But how do you connect it, and how do you make use of the available avenues to solve the problem is what probably you can call as innovation. Thank you for this.
That's
Dr.
innovation.
Bilge, Dr. Bilge, you would like to?
I agree with Neha. Innovation by definition is creating a new idea, but also at the same time making something better, right? You may not create something, from zero, but there's some existing, technology or understanding of the thing, and you can just build on it. It's still, I think, innovative in that sense. And I agree with you that first we accumulate knowledge, and then if we are talking about, for example, drug making, vaccines, or biotechnological devices, these are all built on the the knowledge that we accumulated through research. So I agree with you. Only thing that I maybe stress a little bit more, emphasize a little bit more, as I said before, is everything. It's sometimes people say without technology there cannot be science, basic science. Without basic science there cannot be a technology. There are two. Sometimes people do this separation, which I find it weird, because without technological advancements, we can't do better basic research, right? For example, to be able to see, for example, cryo-EM, right? Cryo-electron microscopy. This was a technological achievement, and people got Nobel Prize in 2018. But without cryo-EM, we now can do things that we couldn't do before. So, and also other way around, right? Without basic research, technology cannot advance. So it's a bit like, which comes first? We don't know, but it goes like together.
Thank you, Dr. Bilge. I think you have actually identified a theme for further discussion about the basic research versus applied research. I think it's all have to be together. It's not that one at the expense of other. Let's start. I think Neha, you mentioned that the current research is relating to cancer, right? Now, the word cancer is always evokes certain fear, certain, the feeling that it is not curable. This is the type of perception which even very educated people would have. Now, I just want to ask you that what is the challenge in pursuing research in the field of cancer? That's one thing. There are many variants of cancer, right? The cancer sort of affects different parts of the body. Now, I mean, what are they different? When I say you're a researcher in cancer, would a different cancer would have different challenges? Do you need different skills? This is where I think, can you just throw some light on this?
Okay. So like when people say we work on cancer, so cancer is, I think it's many diseases in one. So to define cancer is just uncontrolled growth. So few cells in your body, they just become greedy and they just want to grow and grow, and they form a tumor, and they become so many and so big that other functions, the normal tissue cannot function, and that causes a problem. So, and that is also one of the biggest challenges in cancer. So the thing is that these cells are so adaptable and they evolve. They're so quick at adapting that it's hard to one to study them because they're changing. So if we sequence them, we can only sequence them at a time point. Like now we have techniques and technologies to understand how they're evolving over time, but it's still like a lot of things like how they change, because my area is metabolism. So how their metabolism changes over time is still hard to study because they're continuously adapting. They grow. So for example, if you know about metastasis, so a cancer that starts in one tissue, for example, you have like someone gets a tumor in lung and then it goes to their brain. So these are lung cells which were growing in lung, but now they have gotten the ability to go into the cell, and for a normal cell it's harsh. They would die. Like normal lung cells wouldn't survive in blood, they wouldn't survive in brain, but cancer cells can. So they're very strong and very adaptable, while our normal cells aren't. So that's the difficult part in studying cancer, I think, like how they evolve. Now when I come to skills, like you said, so I think if you are studying one type of, so a lot of people study more than one types of cancer. Of course, you need the domain knowledge. For example, breast cancer would be very different from lung cancer, but the techniques are the same. So you don't need to get additional skill. So you can sequence all kinds of cancer types. You get similar kinds of data. The knowledge is broadly the same, but specific domains, so specific mutations for breast cancer or specific mutations for lung cancer, that you gain, I think you can also gain during your project. So once you start working on something, you start looking at breast cancer data, you read something, and then you start learning about it. Yeah, so I think it's not special or
Yeah,
strange that people
yeah.
work on more than one cancer types.
Thank you. I think you explained nicely, but I think my interest is to see that means we should be able to learn by documenting the research in one particular cancer to the other one. But I think, I don't know whether you ever thought about it. If the cancer cells can adopt itself and survive anywhere,
Yeah.
after all, the cancer cells are created inside the body. It's created in our own body. I mean, in one's body. Then you just see how it is adopting, whereas the other cells are not yet to adopt. Well, this is a research which you need to think about it. I think it's yes. I think expertise available in one domain of research would lead to solving problems in the other type of cancer. Is that what I understood? Thank you, Dr. Bilge. I think. I've seen that you have gained a lot of expertise in design and interpretation of clinical trials. Today, in the era of COVID-19, a lot of things are being talked about clinical trials. And how do you have clinical trials for the vaccine or even repositioned drugs? So can you tell us about what goes into conducting these clinical trials, the challenges one faces, and skills one requires to pursue?
So clinical trials are to test whether the drug that you propose that is working to solve a disease is working or not, right? So that's what, in general, what clinical trials are trying to achieve. And they have 4 phases. So usually, not usually, all the time, phase 1 is testing whether the drug, I mean, it can be small molecule or we call them biologics, which is protein based, whether it is safe to administer or not. Because you may have a drug that is working very well in the lab, but once it is administered to people, it's actually toxic. Then what good you can have it, right? So first phase is about testing this, whether it's safe or not. And if it is, and it is usually being tested in small number of people. And second phase is, if you go to phase 2 in clinical trials, it's about, again, you are testing its safety, but also you now compare it to the standard care for the whatever disease you are trying to solve. So if you pass phase 2, which means your proposed drug is safe enough and shows some efficacy and is better than the standard care. And phase 3 is about a long-term efficacy. So you don't test safety anymore in long-term, whether it is really working or not. And phase 4 is usually a long-term effectiveness of the drug. And in every phase, actually, it's better to enroll more and more people to your clinical trials to have statistical, power. Especially at phase 4, for example, you need to test this in like thousands of people to really be confident about whether it is ready to be, administered or not. Basically, these clinical trials are being conducted by big pharma because they are expensive. You need to, enroll people. So what one can, if you are working in a big pharma, you need to first know about the regulation as well. And it depends where you want to, start selling that drug. So we always, when we say clinical trials, we think about the United States because mostly that's the biggest drug market in the world. And you need to know about regulations in the United States, FDA, what FDA requires to do to be able to, pass these phases. But if you are working in India or in Europe, they also have their own clinical trials. So that, that more or less they are similar, but there might be small differences that can change the outcome of your clinical trials.
Yeah, thank you. But I think, let me ask a very uncomfortable question to you. So today we are suffering with COVID, with so many people's lives are gone and so many people's lives at stake. Do we have an option to reduce the time that is required for completing the clinical trials?
Yeah, I think FDA has a special path for, in case of pandemics or emergency uses that can shorten this clinical trials. And for example, one of the drug that is, I think in May started to be used in the United States was remdesivir. So it's already, it's in phase 3.
Yeah, no, I think, repositioning of drugs is one thing, but clinical trial for a vaccine is a different ball game, I think. But what you said is that there is a provision to reduce the time. That's a good answer to people, like many people who are looking for the solution to these problems. Yeah, let me switch from bioscience research to life, and then let me ask Neha. Neha seems to be uniquely placed. She's from India. She both in North and South. Interesting to see that you studied at BIT Vellore.
Yeah.
Went to Iceland to do PhD from hot to cold woven, I think, and then and now you're in Singapore. It's a very diverse will be there. Yeah, I think so. If you say Singapore is hot, I don't know what will you say to Chennai, and in Vellore, what you would say.
No, I mean it's same, I would say.
It's similar. Yes. Now, I think I want to ask you that how different the environment was. When I say environment, go beyond the temperatures. I think the environment was there, and what takes to adjust. This is the time when people start going to different places, adopt it, and excel. I mean, what is your take on this?
Okay, 1 second, Neha, before you answer. Okay, for the want of time, let us keep it very short, the answers, and let's move on to the Q&A session. So, yeah, Neha.
Okay, so I think there are, so when you move to, like, I have moved many times. I grew up in the north of India, then I did my master's in the south, and as you mentioned, like, then I moved to Iceland, which is similar to Europe, like Northern Europe, Nordic. Then I came here to Singapore. So I think like when you move to a new place, it's very similar. The experience every time is very similar, at least for me. You go, there are new people. The culture is different. The working culture actually, the culture, the tradition affects the working culture, the working environment as well. But most of the times these days, it's very multinational. There are many people from different countries. Like for me in Iceland, there were a lot of Indians, some Chinese people, some European, some American. So it's very international. And when the group is very international, you get to know about these people along with working. And I think like I say a lot about culture and tradition because along with work, I think once you try to learn about the culture, it's easier to solve or resolve work related conflicts, or you form a better team, I would say. So to adopt, I would suggest, like if someone is planning to move to a new country or a new place or a new work environment, I would suggest just like go there, be honest with your work, because I mean, of course you're going to work, but try to understand life beyond work as well. So make friends, talk to people, and learn about their traditions and cultures, and just have fun. And I think
good, Neha. I think so. Understand the culture and tradition. That's important. I'm sure you would have done it when you came to Vellore from Delhi. I guess that did. Okay.
yeah, the food was very different also.
Yeah, I know. I think. Let me ask. I think since Viveka is telling we are running out of time, let me ask to Bilge a different question. This is about cholesterol levels. You touched about your cholesterol, which you mentioned. Cholesterol is made only in your body and not from outside. You can get it. In a lighter side, in the colloquially, when in India, when the mother tongue, they say, if somebody is arrogant, they will say, is it kove kuvainda? That means does the cholesterol is more? Typically, it'll be they say like that. Now, what I want to understand is that, yes, I think everybody is worried about this high and low cholesterol in the blood, which will affect our health. Now, I think how do we attend to and what direction the treatment is going to see in the future? Because since
that's a good question.
Since you said in your introduction, which I learned today, is that cholesterol is made in the body, cannot be made from outside.
We can take it from diet, and also our cells also.
I think it is.
So in the current standard of care for people who have high blood cholesterol, bad cholesterol, LDL, is they are prescribed statin as a cholesterol reducing drug, which is affecting basically its target is a pathway in the biosynthesis of cholesterol. So you are basically reducing the cholesterol that your cells make, and that's why your cells would like to get more cholesterol from your diet, basically from the blood. So you are reducing your blood cholesterol levels. But in the future, actually, future is happening now, is there are companies that come up with techniques that not techniques, but treatments that uses CRISPR. CRISPR is I think maybe it's just, the basic research that described CRISPR came in 2012. And we are in 2020, and in 8 years, it already has become the standard of deleting genes in the research labs. But now people want to, move one step forward and move with this CRISPR technology to clinic, right? So one of the company that caught my attention was to use this CRISPR technology to delete one of the gene in your body that makes cholesterol, but it's not totally removing, because you actually need your cells to make cholesterol, but it will knock down, we call them deleting the gene that can help to reduce cholesterol. So rather than taking the statin drug every day, you will have one-time treatment if you would like to, and you will not have to worry about your high cholesterol levels anymore. But again, so we have lots of controversy around usage of CRISPR, right? How is it going to unfold in the future? I don't know. But there are companies already trying to move this to clinical trials. So
yeah, good to know. Good to know that
yeah.
we will have a solution for high cholesterol. I think now I have a few more questions, but I think we will give preference to the question answers. And now Viveka would let in the questions, or
Thanks,
so
Prof. Yeah, thank you so much. I mean, we really believe in having time. So, okay, that was a very good panel discussion, but I see there are a lot of questions coming up on the chat already. Harshini, can you mute yourself? Harshini Devi, can you mute yourself?
can we request the audience who want to ask questions to unmute both video and audio and participate in the discussion?
Yes,
okay?
I'm going to tell that, Prof. So I see questions from Dr. Samuel from SRM. Dr. Samuel is also my very close friend. Sam, do you want to turn on your video and audio and ask the questions yourself, or do you want me to ask on your behalf? Dr. Samuel, if you are still around. Okay, and we also...
You just ask on behalf of me. It's fine. I put the question on student perspective that I used to receive.
I know.
So are you able to see this chat? Or
I think because your audio is on. Why don't you shoot the question yourself? That would be more interesting. Yeah.
Okay. So I'm very glad to join the session today, and I received a very good quantity of knowledge about the present aspects in cancer and other field going on through Neha and Bilge. And actually, I want to put question on student perspective. As I am in a higher education institution, many of the students has now passed the final year passed, but they were in the stake of how to move forward for higher education in abroad institution as of now. The present scenario is concerned. It has a two problem. One is the global problem that and second is that their parents are not again allowing to go. So how the students could be able to tackle is that sometimes we feel that even the laboratories in abroad, they put forth a boundary around them so that like it is safe for use the manpower from around their own country or around the same institution to fill the vacancies. Is that going to happen, or is it possible to again revert back what happened before, like again global collaboration between the institutions? Just put some light if you are well about that same thing.
Thanks, Sam. So Neha or Bilge, any one of you can go with that answer.
Well, yeah, definitely there's a big problem for students who would like to go abroad and, study abroad. But I think I don't have, optimistic answer for that. I think for the foreseeable future, it might be really difficult to go abroad and continue your studies abroad. I'm not saying it's going to be impossible, but it will be difficult. So if my suggestion would be to turn to these online courses that you can learn more about, for example, computational analysis, right? That doesn't require lab work. So anything that requires wet lab work or any, Master of Science, PhD in lab work, that's going to be affected for the foreseeable future. So unfortunately, maybe to be able to survive, you need to change your strategy and you need to think about what can I do in these times, which is maybe learning a programming language that's maybe helpful in, computational analysis of, biological things. That will be my strategy. Yeah, maybe Neha would like to add.
Yeah, I mean, I agree with Bilge when she says that you can, these days, like online courses there are many online courses, and universities and institutes have started accepting these as legitimate courses. There reputed universities from the US who have their courses, their masters online. But of course, these are all only for software related things like computational things, programming, data analysis, which is going to be big though. Like
Yeah,
post
definitely.
pandemic data analysis is like there's tons and tons of data that needs to be analyzed, and it's being generated nonstop. So I think and people have started realizing data analysis is very important and it gives insights. Data generation, of course, is important, but until travel bans are lifted, it's hard to say.
But I think let me not let me interfere and then say that Dr. Sam, I think we should say we are in a pause mode now, in a sense that for a few months of pause. And I think what is important is that some of the very premier institutes have started remote campus operation. I can be in Chennai and study at Harvard, or I can be in Chennai and study at NSU. That would happen. But I think I agree with all of you that, the way we learn things has to be re-looked into it. This is what I think, when you start let us say I start a master's course, right away, don't get into the lab and start doing things. I do go through certain coursework, certain do things. So this would happen. So maybe you have to reprioritize your learning and then wait for maybe one semester. That's the problem, Dr. Sam. It's a problem in SRM too. I think, what are you going to do to the students when they join? It's a problem.
We sorted out
So I think what would happen is, it's a problem which all of us are worried, those who are especially handling the students. But the hope is that today we are inventing things which were reluctant to do before. The earlier, the learning is only by going directly there. In fact, see, today we need to change the way we think about learning. I think, the learning is what I think it helps us in a better way. Sometimes the crisis will find solutions which you are hesitating to take it before. I think we can debate. This is a thing which we need to. So maybe Viveka will go to the next question.
So we only have 10 minutes of our stipulated time, but audience, I should just remind you, today we are going to be exceeding by 10 or 15 minutes because we also want to summarize. Okay, one more thing to the speakers and moderator is that we have so many questions coming up. Audience, we might not be able to cover all the questions now, but we will try to answer it very crisp so that we can cover as much as question possible. I have two questions from Prathamesh. So Prathamesh, do you want to come forward yourself and ask? Prathamesh, if you're here. Oh, hi, Prathamesh. Feel free to ask yourself. Thank you for asking the questions though.
Hello, respected speaker. Myself, Prathamesh, and my graduation is completed in agricultural biotechnology, and in future I plan to do MSc in biophysics. So can you please tell me the scope and recent researches going in this field?
Okay, thank you, Prathamesh. Neha or Bilge, any one of you can answer on the scope of biophysics for Prathamesh and for everyone's sake. Thanks, Prathamesh.
So I think I have some experience in biophysics, so I can start, then Bilge can add. I did some biophysics during my PhD. I used some biophysical techniques. So I think, like we have discussed earlier, it's just about the technique. So during your MSc in biophysics, you will learn some concepts and some techniques. And I think there's a lot of scope in the future where you can apply it. So you can change your field from agriculture right now. You can go into medicine and biomedical sciences or biotechnology and apply those concepts and techniques to understand different kinds of systems or understand different systems, biological systems.
I agree with Neha. I think biophysics, it's what I am doing, and actually some part of what Neha is doing, I'm sure is having biophysical aspect. So we can't separate biophysical aspect of the research that we are doing. For example, understanding binding energy of a protein to a ligand, let it be lipid or drug. This is all under the roof of biophysics house, right? And the techniques, as Neha said, if you get the master's degree in biophysics, probably you will learn the concepts and the techniques that people use. But it's I think very prospective, and yeah, I will definitely think about it.
Okay, thanks
Yeah,
both of you.
yeah, just want to add to Prathamesh. I think this is the times when specialization to develop expertise, but application in any field that can happen. I think so. Do not worry about whether I should specialize or not. The days of specializing too much, I think, are going to be in the past. Now you need to apply them to other plus. Yeah.
Yeah.
Okay.
Okay. Thank you. Thanks, Prof. Thanks, Prathamesh. So Nityashri, do you want to come and forward and ask the question yourself? Yeah.
Yeah, sure, ma'am. Can you hear me?
Yeah, we can hear you.
Okay, ma'am. Hello, everyone. And I have a main question. Can you please explain about the field of epigenetics and what are the importance in epigenetics?
Okay. Who wants to answer that?
Explain epigenetics. Well, epigenetics tell you, for example, you have genome, right? Set of genes that express proteins, and proteins do the job in your body. Epigenetics, on the other hand, tells you which gene will be switched on and when. So it is very important, because actually, even though you have the same genetic material in every cell of your body, not all the genes are turned on, right? Some genes are turned off. That's why it becomes, let's say, neuron cell in your brain. But some genes, other genes are turned on, and it makes you to have lung cells. So definitely epigenetics it has been a very fascinating field for the last maybe 10 years already. So, and it has an implication on now anti-aging field, because we now know that as we age, when actually some of your genes turn off, normally they should be turned on and express the protein that your body needs, but with age it is changing. So if you can understand why it happens and what are the factors that makes that gene turn off, which is part of the study in epigenetics, yeah, you can understand aging more. Yeah.
Okay, thank you. Thanks, Bilge and Neha. So, okay, audience, you can put your question on chat. If not, Kanmani has requested me to ask the question on her behalf. Okay, Kanmani asks, is there any career perspective for scientific writing as a career? Yeah, that's a question.
Yeah, actually, because I remember from when I was applying after my PhD, I came, so which was 3 years ago, scientific writing was becoming more and more popular then. So it has been growing since then. So I would say there's a lot of scope in scientific writing, because you see, like now you need to, so everyone cannot read scientific papers. They're just like very dry with a lot of scientific verbs and words and data. So to communicate that scientific knowledge to common people who do not who are not from science, who can be in finance or who can be in other fields, but they need to understand whatever knowledge is being generated in science. So that's why scientific writing is important, and it's growing since then. So I think it's good career choice, I would say.
Yeah, Kanmani's question prompted me to get in. I think
Yeah.
This one domain which currently I'm concentrating. I think no, what is important is, one is to develop expertise in a domain, and other is to express it. And this, I think we discussed in our first session about this part of it. I think it's very important to get domain knowledge and then start interacting with people, and when expression would follow it up. I think generally what would happen is, you become a scientist or a researcher. At some stage you realize that maybe you're good at writing or good at communicating, then do a shift to that domain. I think that should be the method. But doing PhD is also learning how to communicate your research. I think this is important. So it's a continuous process of learning, and then you would take out. At some stage, you decide, okay, I love this more, and then I do that. Yeah.
Okay, Jayashree, can you put yourself on mute unless you have some question to ask? Jayashree, okay. Audience, any more questions?
I have a question to ask.
Yeah, Akash, shoot.
Yeah, so do you see some tools like AI giving us a fast track, especially with clinical trials? Because clinical trials, there hasn't been much improvement since clinical trials has been established. Like, do you see some tools like AI giving a boost uh, to getting drugs faster?
Yeah, not AI, but there is like a computation biology company, Schrödinger, who now partnered with Google Cloud to use their cloud system to do calculations, millions of calculations to find drugs, small molecules that that can, for example, bind to COVID-19 spike protein. For example, it's like drug development, fast and speed up drug development. That's one example. But there are related to AI, for example, again, Google's DeepMind company now come up with AI, some AI algorithm that can predict structure of the protein. So I'm not sure, are you familiar with the crystal structure of the proteins? So we need to spend time, like years maybe, to find the structure, crystal structure of a certain protein so that we can, do screening for small molecules that can bind to it, or you can even co-crystallize this. But now this DeepMind can, with using its AI algorithm, can find the structure, solve the structure of that protein. So you are basically removing, and it's really, the accuracy is very high. Without doing an experiment, now AI can find that. So it's definitely a fascinating era is coming. But we need to find the common, ground. Otherwise, experimental scientists will be like out of job, maybe. Just joking, yeah.
I think.
No, I would like to add there. So I think what Bilge said is true. So AI can be used to predict the targets, but we will still need the scientists to validate them.
Yeah, right. That's why I said, just.
Okay. Thank you both of you. And we have a hand rise from Dr. Tushar. I think we had him last session as well. Thank you so much, Dr. Tushar, for joining us on all our sessions.
Hello.
Good
morning.
Good morning.
Actually, it's a good attempt that you people are doing.
Thank you.
It's giving too much of information. So I'm also a biotechnologist since last 20 years. I'm teaching, doing research in the biotechnology. So just a question which comes to my mind, that when this pandemic started, there were thousands of papers which came out, maybe in biotechnology or in bioinformatics. So how do we correlate the authenticity of such kind of research to get a particular product? Because we are trying to get the vaccine or some medicine against this particular coronavirus. And there are thousands of papers, as I told you, they are coming up. So how do we correlate the authenticity so that you get some good result out of it? So that is one question which comes to my mind, if they can throw some light onto it.
Okay, thank you so much. It's a very wonderful question.
Okay, I think a lot of papers when this started, especially the sequence of the virus, they're published on bioRxiv. These papers are not peer reviewed. I mean, it will take some time, but because of the pandemic, it just needs to be done fast. I think what can be done to correlate is a meta-analysis. So what best can be done is take, say, like 10 papers and see if your results correlate with like all of them, or at least some of them. And if something is contradictory in, or like 50% papers say this and 50% papers say something else, then it's just, you have to take it with a grain of salt. And if everyone is agreeing, then it's true. I think.
Sorry, I want.
Yeah.
Yeah, sorry, Neha, complete.
No, I was just saying it's just because
I think I'm tempted to. I'm tempted to get in. Dr. Tushar, this is where the AI would help us. A lot of information is there. well, I think it is good to have a lot of information. I think this where I think the question previous was how AI is going to help us. I think there will be some firm putting all this into one place and say, well, this is it, and that's not, because there's no time for us to wait for them to be analyzed. And in fact, this summarization is where AI would help a lot. This data analytics. I mean, I would call it as data analytics and say, well, this could be the potential, so that you don't have to repeat those things. I think this is where I think the information scientists would play a role. I know that they would say, yes, this is what could be the solution. It, in fact, the information becoming pandemic is a problem we are facing, and that's where I think the tools like data analytics, data for the forecasting, analyzing, these things would help. And then if you are smart, you don't pick up wrong thing. I think you would like to do.
Yes.
Okay. Thank you, Dr. Tushar. Thank you, Prof. Okay, I think we can kind of come to the end, because otherwise we might lose the audience. So may I invite Prof to kind of summarize today's session and also the past 3 sessions. That's the bonus for the audience today. Without even attending the previous sessions, they can know what happened then.
So thank you, Viveka. I think you all agree that we have a very good discussion with a lot of questions still to be answered. In fact, I had some questions to put to these panelists, which I held because of giving relevance to the question. So you agree that, I think it's what the takeaways for me is that, what Neha said is that if you want to go to a new place, learn the culture and the traditions of that place. I'm sure both Bilge and Neha traveled. Bilge coming from Turkey and then coming here, Neha going around the world and coming to Singapore. This is one takeaway. I think it's important. And then the other thing is that what I learned is that doing research in cholesterol is very important for health, and there are people looking into how you can address this. I'm sure the CRISPR would take us forward, though there are ethical issues we should worry about in CRISPR. I think we need to really, it is a topic which need to be discussed separately, but there is a way out to solve the CRISPR problem. I think, the cholesterol issue, which is, when you become a little old and you realize that, these are all worrying things for you, your cholesterols and things like that. I think, so the cancer is what really appealed to me is that, a cancer research in one organ would continue to the next organ. I think, it's not that, when a cancer affecting a particular one is probably there can be a lot of similarities across the domain. I think so. That means there is a lot of similarities. This is where AI can help us and say, okay, this organ, this cancer, this is the research done. Maybe you can learn from it, and then you can go. I think that's another takeaway which you can get from this. I think we need to. These two researchers are examples of how you can adopt to newer challenges. So I think what impressed me is that what Bilge told. Her academic credentials say she's good in polymers, but her heart is with bio. And you could see that she could come through that, and she finally landed up doing bio. I think it's important to be focused. It's a battle between mind and heart, and what you're good at it and what you wish to good at it. I think one has to recalibrate about this on a continuous basis. This is advice to all of us. I think, it's not that one time I decided and that's it. It's possible that your polymer background would help you a lot when you're looking at the cholesterol. After all, the cholesterol would be a polymeric molecule. I think it would help you to do that part. So a lot of takeaways here, a lot of interest in the audience. I think today's discussion was lively for me. I think it's nice that if you're pressurized for time, that means you're doing well. I think that's what I would say. So I would say that, very briefly, I think I would like to summarize what this series has done.
Prof. Before you summarize, I think a few of the participants are leaving, so you can just make it very crisp. Participants, I really encourage you guys to stay back. Please don't leave for your own good.
A minute. I think it's a series started on 14th. The first session was on implementing and communicating innovations. Today, some questions were about how do you communicate and how do you implement. You have to get an innovation. What is innovation? how do you implement and how do you communicate? And I and Agatha were the partners, and then Viveka did the job of hosting and moderating it. And we had an interesting discussion is about, I think Agatha sharing the challenge in implementing, and I talking about the importance of communicating. That has been the first series. The second one is on innovating and commercializing inventions. In fact, the discussion what Viveka led was, how do we commercialize? You know, I may have an innovative idea, but it's important to take it to commercial level. That is, take it to the level. When I say commercial, means take it to the level where it is being applied, where it is being used by the people, and how that is the most important thing in all innovations, that it is being used by somebody there. So I think it's the journey from being a researcher and taking the research to stage of commercializing was discussed. And both Marlena and Viveka shared their challenges faced being women, how the journey has been. I think they were passionate about sharing the challenges which they had. In the third one, we talked about the data-driven world, and the data, it's a data everywhere. We talk about both Hemanand and Shivam talked about it, and they shared the journey of becoming a data scientist. And I think what is becoming a business analyst and how data science is changing the world. I think that's what was explained, and how it's going to impact the R&D in biosciences. I think this is something which was discussed, because many of you would may have been coming from the biosciences background or the health sciences background. I think it's been personally, I enjoyed interacting with the people and being part of this initiative. The question asked by Kannan today encourages me that, you know, this it required to bring clarity to people about doing research, and communicating research is a challenge, and we need to do not separate them. It has to be, I think. So Viveka, I try to be brief. I think we need to. This is like, and I'm sure that when Viveka would tell, all the recordings are available on the website, and also on the Facebook, and maybe on the YouTube. And those who missed, they should be able to visit those and then go through it. So I would say that, you know, one of the uniqueness of this web series, this is for Viveka and also the audience, is that the panel members are young researchers, young and aspiring researchers, who have just taking off, you know, in the exponential curve. You would see the takeoff situation is there. They are students and also researchers. I think it's a very unique way of selecting these people. As you become a student, your approach is going to be different. And once you become a researcher, established research, your approach will be different. I think what we need to understand is at that interface of making the transition to an established researchers. So I'm taking the liberty to tell you are in the process of becoming like that. I hope it is not, you will not feel bad about this statement, but I think it's unique in that sense that, you know, you are sharing your journey, your experience. This is true for all the speakers. Okay, I can be an exception. I think this is what Viveka mentioned in the beginning. I'm in the other side of the journey. So I guess it's good to be. I know I always had the privilege of being with youngsters all the time in my career, because I was heading the training school with a lot of research scholars around there. So it has been a pleasure. I'm sure that such efforts. In fact, the title is the web series title is about in the new norm, innovations in the times of crisis, stepping into a new normal world. We have already stepped in, and then now got connected with a lot of people. And this is the new norm, and I'm very strongly convinced that this is going to open up newer opportunities, newer collaborations, which would lead to homogenize the world. You know, today it need not be restricted to one region now. I think it is spread. Already the internet, social media brought that, but this compulsion has brought it here. So let's appreciate Viveka for the initiative, and appreciate all the speakers for their time, and audience for being there. And now, Viveka, you would have the.
Thank you so much, Prof, and thank you so much, audience, for staying long. We are almost towards the end. My special thanks to Prof. Sai Baba, because he's the only man besides me and Hemanand who's stuck through the whole thing and was always behind and had our back in making this webinar series a success. So really, really special thanks to him. He's an amazing guy. If you have any queries or any career related stuff on science communication or policy making, please, he is so approachable. That's the thing. Having done that, he's such an approachable professor. So please do write to him. And special thanks to our speakers for today. We really had very good audience, and the speakers are two of my very close friends who are also as passionate as me in trying to share their journey and also career perspectives. If you guys want to write to them, and I will be sending. I see some requests to send all the webinar links. Definitely, I'll be sending. And I also want to specially thanks a few people who have been there for me, like online, offline, like Dr. Samuel of SRM. He's been very steadfast in organizing the webinar on the SRM side. So special thanks to Sam. And also today we have Professor Uday Suryan, who was the HOD of Biotechnology for or TNAU. So he has joined. He wrote to me a month ago telling that he will join this, and he remembered that and he joined. So thank you so much, sir. And last but not the least, as always, we have Dr. Jnanam, who is the HOD for Bioinformatics in TNAU. She is that one woman who always inspires me, and she's here today as well. She was there yesterday. And if she is around, can I ask her to say a few words? Ma'am, Jnanam, ma'am, are you around?
Yeah, sure, Viveka.
Yeah, thank you, ma'am.
Yeah, please unmute your video.
I think I'm at home.
Okay, ma'am, no problem. Just audio is okay.
Yeah. Okay. It's not about a kind of vote of thanks, but I would like to share my opinion on the series of the kind of presentation or the panel discussion we had starting from 14. I would say all the four were excellent. And really, I congratulate and appreciate Dr. Viveka of Avantgarde Initiative for doing this wonderful thing. And as mentioned by Dr. Sai Baba, all the panel members, at least I can say majority of them, they were young. Really, that will again inspire the young minds, because, generally panel discussions we used to have with the experienced speakers, and based on their experience, we used to have this. This is really a very nice kind of arrangement what you have made. I would also like to thank Dr. Sai Baba because, all the panel discussions were very exhaustive and very critic and very sharp. And as you said, he could be able to get all the information from the participants in a very nice way. And so congratulations, Viveka, and I wish that you should do more and more.
Thank you. Thank you so much, ma'am. Thank you. So
yeah, thank you, Professor Jnanam, for nice words. I think, yeah.
Okay, so very, very last leg of the program, right? As I promised earlier, so we are trying to do a mentorship program partnering with NUS MBA. It is for people like you, audience mentees who want to uplift their career, their personality, their English speaking skills, whatnot. You will be paired with a mentor from NUS MBA alumni as well as current students, with whom you can try to build a rapport over a period of 3 months and even beyond. Try to have one-on-one discussion. All this is for free. So I know most of you have registered with me, so I will be sending out the brochure and registration link along with the feedback link for today's session. So please feel to fill in, and the program itself will start somewhere around August. So that's for the 38 participants who decided to stay till the end. So that's kind of an added advantage. And yeah, if you want to know more about us and the past link, a link to the past webinars, then please log on to Avantgarde-Initiatives. And if you have more questions, I will reply if you email to avantgardeinitiatives.com. Again, special thanks to Prof. Sai Baba and the speakers today, Hemanand and Akash. Also, I should mention, because Akash was an audience in one of our very first webinars. He turned around and he's now a part of Avantgarde Initiatives itself as an executive. So I encourage people in the audience today to come on board and join us. We are all working on pro bono basis, just to remind you. So that is it for today. Thank you so much, all of you. Any questions, please feel free, and you can all leave now. Thank you.
Yeah.
Thanks a lot. Yeah.
Okay, stay safe and take care,
Yeah.
all of you. Yeah.
Sure.
Yeah, thank you.
Thank you.
Thank you very much for
Thanks, Bilge.
moderating, Prof, and hosting Viveka, initiating such an important, meaningful webinar series. I hope we were, helpful in one way or another.
Of course
Yeah.
you were. Yeah. Thanks, both of you. Yeah. Okay. Bye-bye then. Yeah.
All right.
Can leave. Bye.
We can leave? All right.
Yeah, you can leave. Yeah, bye-bye.
Okay.
Yeah.
Yeah. Hemanand will end the meeting.
Yeah, Hemanan?
Yes, I think, Viveka, I think you need to share the feedback form later.
Yeah, I'll do that.
Email
Yeah.
address.
Yeah, I'll do that. I think we can end the meeting.
Yeah, all right, I'll end the meeting. Okay, thank you all participants. Have a nice day.
Nice interact with you, Hemanand. I think it's good. I think so. We will continue our interactions. Yeah. Okay.
Yes, Prof. Sure.
No, Prof. Yeah, we'll always stay in touch.
Okay.
We'll stay in touch, definitely.
Okay.
Okay. Yeah.
Yep.
Bye.